for the interruption here. We are continuing with the discussion. Okay, this is where we stopped. Oxidative decarboxylation of isocitrate. So isocitrate is oxidatively decarboxylated into alpha keta glutarate. In the next step, what happens? This alpha keta glutarate, okay, will once again undergo oxidative decarboxylation, okay, as to form succinyl coenzyme A. To form succinyl coenzyme A, where the carbon dioxide from alpha ketoglutarate is eliminated. This step is similar to pyruvic acid decarboxylation. Pyruvic acid undergoes decarboxylation forming carbon dioxide, isn't it? In the similar way, here too alpha ketoglutaric acid will undergo oxidative decarboxylation to produce succinyl coenzyme A, where a molecule of coenzyme A is added and NAD plus is reduced to NADH. Okay, so that means the byproduct of this is the second carbon dioxide that is released in the Krebs cycle. Within the Krebs cycle, 2 carbon dioxide molecules will be released, and as a whole there will be 3 carbon dioxide molecules that are going to be released for 1 pyruvic acid molecule, and for 2 such pyruvic acid molecules, in total 6 carbon dioxide will be eliminated. Okay, in the next step, step 5, this conversion succinyl coenzyme A will be converted into succinate or succinic acid, and here the enzyme used is succinyl coenzyme A, and in this reaction 1 molecule of GTP is produced. This GTP is nothing but guanosine triphosphate. This reaction is a coupling reaction where the GTP here formed will donate its 1-phosphate to ATP. ADP. It will donate it to ADP in such a way. That a molecule of ATP is produced here. A molecule of ATP is produced here, resulting in GTP is converted back to GDP. This reaction is called coupling reaction. Okay, it is called coupling reaction. ADP to ATP, GTP to GDP will be formed. That means actual product formed here in this reaction is 1 molecule of ATP. Directly 1 molecule of ATP is produced, and there is no further release of carbon dioxide. Instead, the coenzyme A that is added earlier will be released in this step. In the previous step, from alpha ketoglutarate to succinyl coenzyme A, 1 coenzyme A molecule is added. Here it will be released so that succinic acid is generated. Fine. Next step, in the next step, succinate undergoes dehydration reaction. Dehydration, removal of water. Okay, so here dehydrogenation takes place. So in this reaction, what happens? Proton will be eliminated from succinate. Proton will be eliminated from the succinate, and here succinate dehydrogenase is the enzyme that is involved. Dehydrogenation. So this proton and this proton or hydrogen atoms are added to FAD. FAD, that is flavin adenine dinucleotide. Flavin adenine dinucleotide FAD is reduced to FADH2. And this is very significant because each molecule of FADH2 can give rise to 2 ATP molecule in oxidative phosphorylation. It can give rise to 2 ATP molecules in oxidative phosphorylation. Remember that. For 1 pyruvic acid, 1 pyruvic acid, 1 FAD. For 1 glucose, 2 FADs will be produced. FADH2 molecules will be produced. Okay. And meanwhile, succinate has undergone change, dehydrogenation to become fumarate. Fumarate. Again, but there is no change in the number of carbon atoms. This is also a 4 carbon containing compound. Next is the hydration of the fumarate again to malate. Hydration and dehydration processes are taking place. So here what happens? Fumarate hydratase is the enzyme, or fumarase is the enzyme that is involved. So now there will be a transition stage of fumarate, that is a carbanion transition state. Carbon ion transition state will be produced, and fumarate will be in this step converted into a malate. Malate. It is a hydration process. Water molecule is going to be added. This is again a reversible reaction. Then in the next step, that is in the final step of the Krebs cycle, dehydrogenation of L-malate to oxalosuccinate takes place. Dehydrogenation. Again, protons will be eliminated from here. So, L-malate undergoes dehydrogenation. That means a molecule of NADH is going to be produced here. The enzyme is L-malate dehydrogenase enzyme. L-malate dehydrogenase enzyme. From L-malate, oxalosuccinate is formed. The question is, here there is a question. See, why all these different changes that happen? Why once 3 carbon atoms are eliminated from pyruvic acid, okay, from pyruvic acid 3 carbon atoms are eliminated, then why the further cycle continues? It is for regenerating the oxaloacetate, so that in the next step, oxaloacetate can combine with the acetyl coenzyme A. Now, I will be very clear about it because we are going to understand this cycle once again, and that time you will be very familiar with this. Now, byproducts of all this reaction is that there will be 3 NADH molecule, 1 FADH and 1 GTP and 2 carbon dioxide that will be released in this step. Coming back to the cycle once again, okay, so here you see we are coming back to the cycle once again so that you will be familiar with this Krebs cycle as a whole. Acetyl coenzyme A, this is the preparatory step where pyruvic acid is converted into acetyl coenzyme A, first step. 1 carbon dioxide is eliminated. Oxaloacetate combines with this acetyl coenzyme A to form citric acid. Okay, citric acid, which is a 6 carbon containing compound. From the pyruvic acid or from glucose, here only 2 carbon atoms come. 1 carbon atom is already eliminated. 2 carbon atoms are there. Out of C6H12O6, okay, we have seen the production of 2C3 compound, that is a pyruvic acid. From this pyruvic acid, 1 carbon is eliminated. We are left with the 2 carbon atoms that comes in the acetyl coenzyme A. Those 2 carbon atoms will combine with the oxaloacetate, which is a 4 carbon containing compound, producing a 6 carbon containing compound, citric acid. Okay, citric acid. Now, the highlighted one, it shows the acetyl coenzyme A that is added here. Okay, the acetyl group of it is here. Now, this will form citric acid in an intermediate reaction. That means this carbon and this carbon are the carbons that were present in the glucose. In the next step, okay, the citric acid, okay, will undergo intermediate product reaction forming isocitrate. But this is a intermediate reaction where isocitrate is converted into cis-aconitase as an intermediate product, where there is a dehydration and the hydration process by the help of aconitase enzyme producing one isocitrate molecule. Those carbon atoms are still present here. Remember, these 2 carbon atoms are still present here. In the next step, what happens? Oxidative decarboxylation takes place, and in the help of isocitrate dehydrogenase, in this reaction, a molecule of carbon dioxide will be eliminated. Resulting in the production of alpha ketoglutarate. Once again, a molecule of NADH is going to be produced. Alpha ketoglutarate then undergoes one more oxidative decarboxylation, resulting in the removal of another carbon dioxide, producing succinyl coenzyme A. This succinyl coenzyme A will then become succinate. By this step, the carbon dioxide that are those 2 carbon atoms that were added are eliminated. Okay, from the glucose. What will be left with the skeleton of 4 carbon atoms? In the beginning 3 carbon atom, and then the rearrangement will result in 4 carbon atom production. So 4 carbon containing compounds are regenerated. In this step itself, 4 carbon containing, after this step it is regenerated. Okay, that means regeneration is complete. the elimination of carbons is complete, but what is necessary after this, once again, sorry, yeah, the ultimately to continue the cycle, this is needed, oxaloacetate is needed. So from succinate to produce oxaloacetate, there will be intermediate reactions, like from succinyl coenzyme a succinate, and then from there the fumarate, and from there the malate will be formed. From L-malate, oxaloacetate is regenerated. Meanwhile, a molecule of NADH and a molecule of FADH and a molecule of NADH will be produced. Okay, simultaneously a molecule of ATP2 will be produced. So this is the whole cycle of Krebs cycle, or whole citric acid cycle. Ultimately, by the end of the citric acid cycle, there will be release of 2 plus 1, 3 carbon dioxide and a few molecules of NADH production. Okay, and all those NADH produced here in this reaction will be then subjected to oxidative phosphorylation, which takes place in the next step of Krebs cycle. And that is all for